Hello there. So basically, I'm going to present about the basics of mitosis and in animals and plant cells. And yeah, you can read this if you want to. I'm not sure if you can even see it to begin with because it doesn't matter if you see it. Anyways, so beginning with first phase, which is well, not really a part of mitosis as it is in cell cycle itself. So I probably should change the title. Anyways, so here is the interface, as you could see, or maybe not. I'll probably describe it because that's probably the easiest way for it to be understood, I guess. So here you have a cell, right? This is a cell, a very oversimplified version of a cell. And basically what it is here's the nucleus, and I mean it's supposed to be the same color, but I put the inner ring inside so that you can actually see it properly without making weird colors. Anyways, so here is the DNA, which is untangled at the moment, meaning that they're probably going to be like squiggling around, all that type of stuff. And in this form, you call it chromatin. Why is that? Don't ask me. I didn't know why they named that. And here you have centrioles and the spindle fibers, which will be useful later in the spindle apparatus or whatever it's called, because I don't remember what it's called. Here you have prophase. Basically, what happens is the chromatins they condense and kind of go into the classic X shape of DNA. Well, not DNA, but chromosomes. And basically in this form, you can draw the chromosome out, like either an X or a really squiggling X or whatever. But the special thing about this type of condensing and stuff is the fact that they actually have a center point where it kind of goes out a bit like a flower, but you can kind of cut it in half into a piece like that called sister chromatids. I don't know why I called that, but yeah, we're just gonna go with it. And so goes with the next phase, which is the metaphase. And as you can see here, yeah, I tried my best doing this with rubber bands because I don't have strings, which probably made it worse. Anyways, so basically, since the nucleus and stuff broke down already, the chromosomes can get lined up in between the cells, and here is what I just told you earlier about the spindle apparatus. Although I don't understand it in full detail, basically the basics are that we have centrioles here, they make the spindle fibers, and those things, well, they have to attach to what they call like centromere or something of a chromosome. And yeah, basically what happens is it's gonna pull out in the middle of the chromosomes to go into the whole sister chromatids that I told you about earlier, assuming you were listening. Anyways, so here's the anaphase. Oh, I should probably align it like this. Anaphase. Basically what just happened is it basically just pulls it out apart, and I just realized I said the word basically a crap ton of times. Anyways, you probably don't really need to know anything other than the fact that after this it's going into the telophase, which is going to be a really quick one, which is this telophase. Now that I realize you could probably not see it because the cell itself is white and black, and the whiteboard is white. Anyways, so yeah, it gets pulled apart into two. How do I say this? Two sides, and what's happening right now is from cell, circle or not, I'm not sure, depending on the cell types and stuff. Basically what's gonna happen is it gets elongated, and then after a while it starts pulling itself apart into a peanut shape, and then after a while into a concave cell and then splits apart, but that's later on. And yeah, basically the nucleus kind of just reforms, centriole splits on two sides. And you might ask me, if this is the end of the cell cycle, then how did you have two centrioles in the beginning? Reason being that this is a part of, well, mitosis and cytokinesis, but it's not the entirety of the entire cell cycle. Basically, what it goes through is G1, just growth 1, where basically the cell kind of just performs normal functions, as synthesis, which is the replication of the DNA using mRNA, RNA, and all that stuff. And then it goes into growth 2, where it starts growing in size and stuff. Well, I mean, all the probably be size. Yeah, whatever. And basically, all of this is just the interphase, which is this one. And then it goes into mitosis and cytokinesis. I'm not even sure if like this mic thing actually works. I've never tried it out, but that's it's good. I don't think it is. Anyways, so yeah, that happens, and now it's gonna revert back to the whole first stage in interphase again. So here I have two cytoplasms to indicate that the cell has been replicated into two cells. But then again, this is animal cell. We're talking about plant cells too. So for that, I also made another one describing about the plant cell. So yeah, this is during the telophase of plant cells. So as you can see, they actually have more like a line splitting in the middle or a line forming in the middle, depending on how you see it. And yeah, these things are basically made from vesicles. If you don't know what vesicles is, they're basically, well, membranes of fatty acids. I guess I don't know how to really like say it, but basically they are piling up, and then because the material is basically the same thing as a cell wall, it kind of just goes in, building up more and more and more, and then at the end of the day, it just kind of becomes a big line of cell wall. And then I started realizing I'm supposed to call this cell plate. So yeah, that's about it. And yeah, that's the end of it all. I explained everything here. If you don't understand, then I don't know how to describe it. Anyways, so for simplicity's sake, I'll explain it by having the whole thing here again, and then I mark which one is which. Okay. I don't even know if you can see on the board. You probably could, or not. I'm not sure. So yeah, telophase. I don't even know if you guys can see the arrows and stuff. But yeah, it's a bit messy because I don't know how to organize my things properly. And this setup took about 35 minutes, and deadline's coming in about 30 minutes. So yeah. interphase, prophase, metaphase, anaphase, telophases, because there are two of them from two different type of cells, and cytokinesis with two cells. I have to write x2 here because there's not enough space for the other one. And that's about it. Enough of me rambling, and this is the end of the video. So yeah, bye, I guess.